an alpha helix across a phospholipid bilayer, describing that as a transmembrane protein, and then extending that idea of a transmembrane protein with its alpha helix into a proper hydrophilic channel. And I'll pick that up again here today and try to describe the relationship between the alpha helices as they form a channel and the three-dimensional structure of the protein as it extends into the cytoplasm, into the extracellular space. But most of our time today is going to be spent on an introduction to compartmentalization in cells, and we'll start there by comparing prokaryotes and eukaryotes. Now, when we talk about prokaryotes in here, we're going to skim over the surface, and we're going to be talking about the eubacteria and the cyanobacteria. We're not going to get into the archaea or any of that. I mean, those of you who have had a microbiology course recognize that I'm oversimplifying this by a whole lot. But what I'm trying to do here with this simple introduction of prokaryotes versus eukaryotes is simply to contrast the prokaryotic biology with eukaryotic biology. This is a course in human physiology, and we are truly eukaryotic organisms, and so most of our time is going to be spent on talking about eukaryotic cells and their organization. But the prokaryotes got here first. They probably evolved within, 700 million years of when the Earth first formed, about, 4.5 billion years ago. It appears now, though, that the eukaryotes came on the scene within a few hundred million years after that. There's a lot in common between the prokaryotes and the eukaryotes. And a lot of those commonalities define life here on Earth. And when we talk about the commonalities, central to that is something known as the Central Dogma. And the Central Dogma is a concept we'll refer to over and over again, but for our purposes here right now, the Central Dogma says DNA is transcribed into RNA, and RNA is transcribed into protein. So we store information in DNA, there's an intermediate step of RNA, and we then generate the myriad of proteins that are important in biology. That's true for all the prokaryotes, it's true for the eukaryotes. There's some detailed differences, the ribosome's a little different, the ways in which the DNA is read is a little different, but by and large, the central dogma of DNA to RNA and protein is true for both prokaryotes and eukaryotes. We find that ribosomes are the protein synthesis factories in both eukaryotes and prokaryotes. So this appears to be conserved all the way back to the initial stages of life on the planet. But there are also substantial differences between prokaryotes and eukaryotes. And one of the major differences between prokaryotes and eukaryotes is this idea of compartmentalization. For the most part, prokaryotes have never figured out the trick of becoming a multicellular organism. They'll sometimes associate in colonies, but they never have figured out how to make tissues and organs and the complicated structures we see in metazoans such as ourselves. And part of that increased complexity in the biology of the eukaryotes arises from the compartmentalization within eukaryotic cells. One of the things that happens in stark contrast to prokaryotes are basically a plasma membrane surrounding a bit of DNA and some proteins, whereas eukaryotes have multiple intracellular compartments, most of which are made from phospholipid bilayers and biological membranes. We're really going to emphasize between prokaryotes and eukaryotes is this idea. So one of the major differences of compartmentalization. One of the stark differences is the way DNA is handled in prokaryotes and eukaryotes. In prokaryotes, the DNA basically sits in the cytoplasm. But in eukaryotes, the DNA is now sequestered into the structure called the nucleus. And the nucleus is really the hallmark of all eukaryotic cells. The nucleus, in fact, the term eukaryote means true kernel. And from the early microscopists going back into the early part of the 18th century, where they first began to see cells, they recognized one of the hallmarks of eukaryotic cells was this structure in the middle of it. They said it's a kernel, and from that we get the term eukaryotic cells. we're going to see that the nucleus not only stores the DNA, not only is all of the DNA sequestered within this membrane-bound compartment, the nucleus, but also we make all of the RNA from DNA. All transcription of RNA into DNA occurs in the nucleus. We'll see a little later when we talk about RNA editing that there are enzymes within the nucleus that also edit the RNA. So DNA is always in the nucleus. RNA is transcribed from DNA in the nucleus, but the RNA then has to go out into the world and go out into the cytoplasm and perform its function of making proteins. So the nucleus now is not just a confined compartment. It's a compartment that has traffic in and out. And much of that traffic in and out is the RNA which has been transcribed and edited in the nucleus, has to be shipped into the cytoplasm. Another big difference that we're going to see between eukaryotes and prokaryotes, and we'll spend quite a lot of time on, is the idea of membrane traffic. Because we have membrane-bound compartments inside eukaryotic cells, we now will use membrane-bound compartments to deliver protein from one part of the cell to the next. And that delivery system uses proteins that are incorporated into something known as a cytoskeleton. It uses motor proteins. It uses membrane-bound compartments as cargo-carrying containers. And we'll talk a lot about that. So, again, the role of the nucleus is DNA sequestration, RNA synthesis, RNA editing, and export out into the cytoplasm through structures called nuclear pores. And we'll try to work our way through this and bring ourselves around to another very important intracellular structure, an intracellular organelle known as the endoplasmic reticulum. We'll spend a lot of time talking about the endoplasmic reticulum. In your mind's eye, you can think of the endoplasmic reticulum as being a bag inside the cell. And we're going to see in some places inside the cell, that bag is going to sequester calcium. In some places inside the cell, that bag is going to be important for making a certain kind of protein, a certain subset of proteins. And in some places, it will be a region that makes lipids. But let's start here with a quick summary of where we were before, talking about the phospholipid bilayer. Emphasizing that there are some proteins that cross the membrane a single time. So they have an amino terminus down in the cytoplasm. They get a certain three-dimensional shape. But when this protein crosses the phospholipid bilayer, it crosses the phospholipid bilayer as an alpha helix. The width of a typical phospholipid bilayer is about 6 to 7 nanometers. Again, don't hold on to those numbers. But if you recognize this as 6 to 7 nanometers and recognize in an alpha helix that the amino acid to amino acid step is 0.15 nanometers, that gives you a sense that it's going to take 40 plus amino acids in this spiral staircase to transit the phospholipid bilayer. Then once this protein gets into the outside world, we'll have its carboxylic acid terminal, and it can fold upon itself and obey the rules of three-dimensional folding that we talked about last time of cytoplasmic proteins. But in this case, we have a single pass transmembrane protein. And where we closed off was trying to think about how we were going to take these alpha helical regions and convert them into some kind of hydrophilic pathway across the membrane. How do we go from this kind of structure to the shorthand term that we talked about before of being a channel, where we described that channel as mostly a hot dog bun or a potato going across the phospholipid bilayer? That happens because we can thread one of these proteins across the phospholipid bilayer multiple times. So again, emphasizing the alpha helix here and emphasizing that there are 40 or more amino acids embedded in the hydrophobic tails of the phospholipid. That means the R groups on these amino acids, to maintain a stable thermodynamic state, for the most part, need to be nonpolar. They need to be uncharged. So in this case, we have R groups that are uncharged. Clearly, if you have uncharged R groups interacting with the uncharged fatty acid side chains, there's no way to generate a hydrophilic pathway through that region. So to generate a hydrophilic pathway across the membrane, we need to modify the structure a little bit. So that the membrane, for a transmembrane protein that's destined to be a channel, it goes across the membrane once, goes out into the extracellular space, comes back across the membrane a second time, goes across the membrane a third time, and then finally across the membrane a fourth time. It appears that the minimum number of transmembrane regions you can have to form a channel is 4. Doesn't mean that 4 always makes a channel. And in fact, many of the transmembrane proteins that are proper channels will contain as many as 4 transmembrane regions. And there are many proteins that cross the membrane several times that aren't proper channels. But what we want to talk about now is how we can generate a hydrophilic pathway from that structure. And this is pretty much where we left off yesterday. And we had numbered those 4 alpha helices as 1 through 4. And all we've done here is to take these 4 alpha helic regions and place them side by side so we can see them. We can label them as 1, 2, 3, and 4. The next thing I want to do, though, is I want to aggregate them around a kind of central channel. And to do that, I want to think about this as looking down on the surface of the membrane. So now, what we're looking at is this surface. This is meant to be a bird's eye. A bird's eye view of the membrane. Again, you think of it one of 2 ways. You can imagine we've taken this phospholipid bilayer and turned it at right angles, so now you're looking down on it. Or you can imagine putting your head inside the board from this view and looking down on it. Then, coming across the phospholipid bilayer will be these 4 circles. So each one of these circles is meant to represent an alpha helix. And now we can number them. 1, 2, 3, and 4. All I have done here is that here we have shown the thread going across the membrane. Now what I want to do is bring these threads together in such a way they're going to aggregate in around a central channel. So if you imagine that you would stand on the number 1 alpha helix and walk down the spiral staircase, the 40 or more steps down the spiral staircase. Remember, every 3.6 amino acids, you go 360 degrees. So you'll go from out here in the hydrophobic region, the fatty acid side chains, around through the channel region and back out again. So there will be R groups coming off of this alpha helix. Now, certainly, these R groups facing the phospholipid tails are going to be predominantly hydrophobic. But now, once we go round the circle, and we go round the circle on all of these, some of these R groups are pointing this way. They're pointing toward one another and the other alpha helices. Those R groups pointing inward in the middle of the spiral staircase. So you imagine these 4 spiral staircases having R groups on the steps pointing inward. These can be hydrophilic. So they can be charged or polar. Well, if you now can imagine these 4 spiral staircases interacting in the middle here with hydrophilic R groups, you now have a series of R groups that are providing a hydrophilic region or a hydrophilic pathway from the outside of the cell to the inside of the cell. And that's all done by exploiting the organization of the amino acids in the R groups and having the charged and polar R groups pointing toward one another to make a hydrophilic pathway. That's how you generate a channel. And you're not confined to 4 alpha helical series. You can have as many, as far as I know, as 24, maybe more. And those all will then have hydrophilic regions pointing inward to make a proper transmembrane protein channel. So any questions on that? Yes?
Does it require at least 4 or more alpha helixes just because it needs to cross the membrane?
So it requires a minimum of 4 alpha helices so that you can orient these R groups inward to make a hydrophilic region, to make it thermodynamically favorable to have hydrophilic R groups pointing one another rather than interacting with the hydrophobic side chains. Yes?
So for the sake of symmetry, does it favor even-numbered collections of amino acids?
Oh, good question. The question here is, does it favor even-numbered to make channels? And the ones I'm thinking of are all even numbers, but that don't make it right. So I think it's possible you could do it with odd number, but I don't know of any cases where you do. One of the places that I think about are the voltage-gated channels, and those are the classic ones that are selective for calcium and for potassium, for sodium. Those are 24 pass. The transmembrane proteins that are ligand-gated receptors are typically made up of 5 different proteins. Each of those 5 proteins goes across the membrane 4 times, and each one of those 5 proteins contributes an alpha helical region to each of those 5 proteins contributes an alpha helical region that makes a channel. So in that case, you would have 5 alpha helical regions making a hydrophilic pathway. But the total number of times it would cross the membrane is 20. So it is possible to have an odd number of, it's possible to have an odd number of alpha helices. But in that particular case, it's coming from the fact that those molecules are pentamers. I don't know if that helps or not, but the symmetry is an interesting question, and I don't think you're held to an even number, but for the most part, I think they are. Anything else? If not, let's move on and talk about prokaryotes versus eukaryotes. Now, when we talk about prokaryotes, the prokaryotes I want to refer to here today are the Eubacteria and the Cyanobacteria. The Cyanobacteria are different from the Eubacteria because these are photosynthetic. They're capable of capturing a photon of light and converting that photon of light by splitting a molecule of water and converting that photon of light into carbohydrate through a long process. So they make carbohydrate by splitting water, generating oxygen from the water, and making carbohydrate. But when we think about these prokaryotes, what we think about is first, they lack a nucleus. They lack membrane-bound compartments. But what they have in common with eukaryotes is that they contain DNA, deoxy, ribonucleic acid, which is DNA. And the deoxyribonucleic acid stores information about ribonucleic acid structure. Now that's a roundabout way of saying that DNA is transcribed into its related nucleic acid RNA. And we'll delve into the details of DNA and differences and similarities between DNA and RNA a little later. But for right now, just recognize that DNA is a storage molecule. It's storing information, and it's primarily storing information about the base sequence in RNA. Then the RNA stores sequence information about the amino acid sequence. In protein. That's a roundabout way of saying the central dogma holds that DNA is transcribed to RNA, and RNA is translated to protein. We're going to spend a lot more time on this, but right now recognize that this is true for prokaryotes. It's fundamentally true for prokaryotes, and we're going to see in a moment it's absolutely true for eukaryotes as well. The other point I want to make here is that prokaryotes use ribosomes made predominantly of ribosomal RNA, and they use messenger RNA made of RNA, and they use transfer RNA made of RNA to work in concert to generate, you can read synthesize, you can read translate, RNA into protein. It's important to recognize this because this is foundational to life on the planet. It appears that life occurred here about 700 or 800 million years after the planet formed 4.5 billion years ago. And then some 600-800 million years later, maybe as few as 300 million years later, the eukaryotes arose. But they use much of the same chemistry. They are based on prokaryotic biology. When we draw an image of a prokaryote, and maybe this is the best way to summarize the fundamental differences between eukaryotes and prokaryotes, is that when we describe the structure of a prokaryote, the structural organization is fairly simple. So if we talk about prokaryotic cell structure, we see that there's a membrane. This is a plasma membrane. Now, some but not all, prokaryotes have a second outer membrane. But if we look into the cytoplasm, we find that within the cytoplasm is a single strand, a single molecule of DNA. Frequently, these DNA molecules are attached right to the plasma membrane, but it's a single circular molecule of DNA. Now, in addition to the DNA out of the cytoplasm, there will be ribosomes. And in addition to the ribosomes, there will be molecules of messenger RNA, and transfer RNA. So this is what's in common with all biology: DNA, RNA, and protein. So in the cytoplasm, there will also be these proteins. And maybe the best thing to hold in your mind's eye when you look at this is what this system lacks. It doesn't contain any membrane-bound compartments. When we begin to look at eukaryotic cells, one of the things that strikes us is the number of different intracellular organelles, the number of different types of membrane-bound compartments that are hallmarks of eukaryotic cells. Prokaryotes have none of them. So let's go over here and sort of quickly make a drawing of the eukaryotic cell. So when we think of a eukaryotic cell, we're going to, again, have a plasma membrane. So there's the plasma membrane. But one of the hallmarks of these eukaryotic cells is the structure, usually toward the middle of the cell, not always, but often, known as the nucleus. And these structures were obvious to the early microscopists. As far back as von Leeuwenhoek looking at cells in 1720. He recognized that there was this structure inside many cells, which was this lump. And he thought of it as a kernel. And so this term eukaryotic means true kernel. But the kernel is, in fact, in biology, the nucleus. I'm going to go up here. I'm going to list some things about the nucleus. The nucleus is a membrane-bound compartment. It's a classic membrane-bound compartment. But it's a membrane-bound compartment that's different from all the rest in that it's a membrane-bound compartment where DNA is sequestered. So let's call this nucleus nuclear characteristics. First, it's a membrane-bound compartment. But it's a membrane-bound compartment that specializes in sequestering deoxyribonucleic acid. That simply is telling us that in eukaryotic cells, the DNA is always confined to the nucleus. But the second major idea and distinction from the prokaryotes is that the DNA itself is organized into chromosomes. Now, it's true that each chromosome is one long DNA molecule. But these chromosomes are characteristic of eukaryotic animals, and what's more, in humans, the nucleus contains 46 chromosomes. The chromosomes are organized as 23 pairs. These 23 pairs, the 23 from the father and 23 from the mother. It's true for all of our cells, it's true for all of us. Those chromosomes that are sequestered within the nucleus are 23 of them came from your father, 23 of them came from your mother. Now, each of the chromosomes is one long DNA strand. And if you take the measurement of each one of these chromosomes, each chromosome may be up to a centimeter or more long and contain several hundred million base pairs. So these are long molecules. They're like a string. They're very narrow, but they're very long. And these chromosomes now are organized around histones in the nucleus. But not only does the nucleus provide the site where the DNA is sequestered. The other point we need to bring up here is that the nucleus is the site where DNA is transcribed into RNA. I should say, too, we'll see a little later, that it also is the site where RNA is edited. So for the most part, whatever you're going to do to RNA, however you're going to modify it, how you're going to edit it, how you're going to make it, that's all in the nucleus. But the other major idea of what the nucleus does is it regulates export of RNA to the cytoplasm. The reason for that is that although we always make the RNA in the nucleus, the RNA functions, I'll say mostly in the cytoplasm. So if you're going to make these gigantic molecules of RNA, which include the ribosomes, in the nucleus, but yet those gigantic molecules are going to carry out their function in the cytoplasm, there needs to be some mechanism of regulating the export of RNA into the cytoplasm, and this is largely done through structures called nuclear pores. So those are the fundamental things that are going on in the nucleus. So up here, coming back to the nucleus, I'm going to put some structures in here, and some lines. And these green structures are meant to be chromosomes. Now, the diameter of this kernel, the diameter of this membrane-bound compartment, is no more than 6 to at most 9 micrometers. So that's large in terms of cellular size, but it's not a very big volume. And remember, what we're going to have to do is we're going to have to stuff 46 chromosomes. We have to stuff 46 chromosomes into that nuclear compartment. Not only do we have to push all of these chromosomes into the nuclear compartment, but we also have to be able to readily access subregions of that DNA so we can transcribe it into RNA and make protein. So you have to not only organize the chromosomes so they fit into this tiny volume, but you also have to be able to access different regions of those chromosomes whenever you need it. It's always amazing to me that you can take upwards of over a meter of DNA and stuff it into this tiny volume. If you're going to do that, you need some organizational framework to stuff that much DNA into this small volume. And the organizational framework occurs by winding DNA around proteins called histones. The histones are DNA binding proteins. And if you look at the amino acid sequence of histones, you find they're among the most conserved of all proteins on the planet. So once eukaryotic biology got going and figured out how to make a nucleus and how to sequester the DNA in the nucleus, it appears that it solved the problem of wrapping that DNA around histones, and the histone structure has not really changed very much since that time. Histones typically, histones are spherical proteins. And they're spherical proteins that self-assemble. They self-assemble into octamers. So that if you have one of these individual histones, it will assemble with a neighbor, and that with a third, and then a fourth. Now, what I'm terrible at showing is three dimensions, and so there'll be another histone behind this. So each of those histones is assembled into a set of 8, called an octamer. And then we can wrap a strand of RNA around that histone. And then this DNA strand will then further wrap around another histone, another octamer. So again, the DNA strand wraps around it. It's not too much of a stretch to think about these histone octamers as being a kind of spool. And imagine the DNA is a thread, and you're wrapping that thread around this spool of the histone proteins. But you can wrap these, and they'll condense into a condensed chromosome, and that's shown here. So in this diagram, what they're trying to describe is how you take a single molecule of DNA. Now, there's a little more detail in this than we've described so far, but the DNA molecule is made of a backbone of deoxyribose sugars, 2 strands of deoxyribose sugars held together by base pairing and hydrogen bonding. That's showing the DNA molecule. This is showing the self-assembly of these histone molecules into an octamer. And once the set of 8 has self-assembled, then we can wrap the DNA strand around that octamer. And then we can further wrap these and coil them until we condense this chromosome. This is a chromosome which has been condensed at just the time of metaphase. This is going to break at this point, and this one's going to go off to this cell, this one's going to go off to the other cell. But having said that, this is an extreme case of condensing the DNA into a chromosome. But throughout life, the chromosome has to be not condensed into this finely packed structure, but it has to be more or less open so that at one point or another, you can access that region of DNA to make RNA. And so there must be some mechanisms to unwind this coil and release the histones and allow for binding of RNA polymerase to the DNA to make protein. You had a question. I'm sorry, say it again? So does it form, so you're saying that the DNA forms a globule of some sort? So the histone, 8 histones come together as this octamer, and you wrap it. And then another set of 8 will be wrapped, and those then will come together. They will coalesce. And these will further coalesce and coalesce. The coalescing of the octamers is, as far as I know, largely dependent upon the structure of the histones. And so then by coalescing the octamers, you're at the same time forming this coiled structure of the DNA and organizing the DNA. Yes, in the back.
What happens to histones during mitosis, meiosis?
What happens during mitosis and meiosis? Well, in that case, what you're seeing here is at the point of mitosis, where these are right around the centromere. They've been concentrated around the centromere. And what's going to happen then is these will attach to a microtubule at this point. And then they will randomly attach to the microtubule, and that's the random assortment period, where you're now going to randomly assort the chromosomes, the chromosomes that will then go to the daughter cells. And that's done through binding at that central part. But that, you're right, that's a peculiar phase in the cell cycle when you have condensed the DNA and have it as much as condensed as possible and organized into individual chromosomes that now can be easily separated during cell division. Does that answer it? Okay. Anything else? Well, if not, let's think a little bit more about what's going on in the nucleus. So we have this nuclear structure. It's a membrane-bound compartment. The DNA is sequestered within this membrane-bound compartment, but it's within the membrane-bound compartment that we are now transcribing that RNA into DNA. And we're transcribing the DNA into RNA. And among the RNA molecules will be RNA that's assembled into ribosomes. The ribosomes are the protein synthesis factories, but they work out here in the cytoplasm. So these are ribosomes. And it's important to recognize that all protein synthesis occurs in the cytoplasm. So we've generated a kind of problem here. We've generated a problem such that we have forced the DNA to always live inside this membrane-bound compartment. We have described a situation where in order to make protein, we have to transcribe the DNA into RNA, but yet that RNA has to leave this membrane-bound compartment and go into the cytoplasm to do its job. That means there must be sites of egress for these molecules to leave the nucleus. And sure enough, at regular intervals all around the sphere of the nucleus are these specialized regions called nuclear pores. Now, this term pores is really a misnomer, because when you think of a pore, you just think of an opening. You think of sort of this open pathway. And these nuclear pores are not open pathways. The nuclear pores are composed of some 300 or 400 proteins. So they're a complex of proteins that use tremendous amounts of ATP to open and close. The way I imagine these things working is they're a little bit like the old portcullis on a castle. If you remember ever playing with castles as a kid, you lower the drawbridge, and then you pull up the portcullis to allow people in across the drawbridge into the castle or out of the castle. I think of these nuclear pores as a kind of portcullis, because you have a lot of energy that goes into opening these things up, and they open up and allow for ribosomes and mRNA and tRNA to leave the nucleus and go out into the cytoplasm. We won't talk about it much in here, but these nuclear pores also regulate the movement of proteins that are synthesized out in the cytoplasm that come back into the nucleus. There are proteins that need to be made in the cytoplasm whose function is with the DNA binding proteins, for example. And they now have to be made in the cytoplasm and come back into the nucleus through the nuclear pores. So the nuclear pores are not just wide open doors. They are doors with carefully controlled opening and closing mechanisms to allow molecules in and out. But that's fundamentally, in short, the structure of the nucleus. If you hold on to the idea that DNA is sequestered there, all RNA is made there, but RNA has to somehow leave this compartment to do its job in the cytoplasm, that's mostly what we're talking about. We'll see a little later that in addition, this RNA that's synthesized in the nucleus is also edited in the nucleus before it leaves and goes to the cytoplasm. So there are a bunch of things that have to happen from transcription of RNA from DNA to its editing before it goes into the cytoplasm. That's all nuclear. But there are many, many other membrane-bound compartments in the cell. And the next one I want to tell you about, and I'll put this one in purple, is there's another membrane-bound compartment here that comes right up against the nuclear membrane. This membrane-bound compartment is known as the Endoplasmic Reticulum. And one part of the endoplasmic reticulum membrane is actually a part of the nuclear membrane. But what I want to emphasize about the endoplasmic reticulum is that it's a membrane-bound bag within the cell. And to sort of envision that, imagine the endoplasmic reticulum as a kind of plastic bag. Imagine that this plastic bag has this wall of the plastic bag attached to the nuclear membrane. But the other wall of the plastic bag is out in the cytoplasm. But there's a lumen inside that plastic bag. There's There's a lumen inside the plastic bag, just as there's a lumen inside the endoplasmic reticulum. And we're going to see here in a little bit that what we're going to do is we're going to attach some of these ribosomes to the endoplasmic reticulum. And then what we're going to do is we're making a subset of proteins that we're going to channel or funnel into the endoplasmic reticulum. And we'll pick that idea up next time.